So we've made it to Unit 2, so we are a third way through the entire National 5 course, and this unit is called Multicellular Organisms. So let's have a look at what you're going to be covering in this unit, and then we'll just get started. So in this unit, the first key area, so the one we're going to be covering half of in this video, is called producing new cells. So the idea is we're looking at how cells manage to make more copies of them, and we'll be looking at a special type of cell called a stem cell and how it manages to keep us alive.
The second key area is split into two parts. It's all about control and communication. The first part is actually the nervous part, which is looking at the brain and the nervous system and how reflexes and things work. And the second part is looking at hormones and looking specifically at blood glucose levels and how we regulate them.
Key area 3, nice short one, it's reproduction. So we're going to have a quick look at reproduction in animals and then reproduction in plants.
Then it's variation, so we're going to look at inherited traits and how we predict certain inherited traits from family trees and things.
And we're going to have a little look at transport systems in plants, basically with a focus on how does water get from roots up into leaves.
Then we're on to transport system in animals, which is all about how the blood goes around the body, basically looking at the heart and all the different arteries and veins and capillaries.
Then we're going to be having a look at absorption of materials, and this is with an animal focus. And we'll be looking at basically two areas where stuff is absorbed and the features that those two areas have in common. So it's a relatively big unit. Again, consult your PLPs regularly as you're going through this. Lots of new vocabulary to learn, but what's new? It's biology, it's words. And yeah, let's get started. So producing new cells is this key area. And so it's key area number 1, and it's about making new cells, funnily enough.
So this is the course specification straight from the SQA for what is coming up in this. In this specific video, we're going to look at A and B. The things to point out really, really importantly, this is that it's the understanding of the terms and what the terms actually mean and how we use them, because that is where you're going to get or lose your marks in this one, is can you actually use the words and say them very specifically in this. So a really, really crucial one to be getting your definition down for. And then C and D we will look at in the next video.
Okay, so overall we'll be looking at mitosis. Then the second video is about stem cells and cell specialization and hierarchy, which that should take about 10 seconds, that last bit to cover. It's quite a small area, even though the words seem kind of large and scary.
Okay, so properly for this video, starting on the knowledge, we're going to just be looking at the process of mitosis, what it is, what happens, why it's important.
So things you should be able to do. You need to be able to name mitosis as the process through which cells are going to divide. You have to describe any of the stages of mitosis. This might be you're given a picture and you're asked what's going on there, or you're given a picture and you're told what's going to happen next. You'll have to identify the stages of mitosis in pictures, there you go, and use these terms, chromatid, equator, and spindle fibers correctly when describing mitosis. There's another couple of language points as well that you need to be familiar with as well, but we'll go through those when we get to them.
Okay, so just recapping on the stuff that you already know, you should know the idea humans can grow and repair themselves. Obviously, that's how you've gone from being this tiny little baby to a grown human now, and how you fix yourself when you've been hurt. All organisms are obviously made up of living cells. We are alive, that's kind of in the name. And the nucleus of a cell contains the DNA, and that DNA has got all that important information which makes you you. And that DNA kind of coils up and condenses into structures that are cross-shaped structures called chromosomes, and they are the things that we're going to be really focusing on today.
Okay, so cells divide through a process known as mitosis. Okay, the mitosis will produce two daughter cells from one parent cell, and the idea behind it is the daughter cells are identical to the parent cell. So what we can see in the diagram below is we start with the cell on the left, and the idea is it has two single chromosomes inside of it. That's what that red and that blue structure is representing. And at the end of the process, we end up with two cells on the right that are both identical to the starting cell. Okay, now that is a very important point to know about, is the idea is the number of chromosomes in the parent cell, sometimes known as the mother cell, is going to be the same as the number of chromosomes found in each daughter cell. Okay, so it's a very important point to know about that.
Okay, so this is a kind of question, it comes up once in a blue moon. It's not too common, but the idea of it kind of comes into a lot of questions, just knowing how these work. It's a really nice, easy numeracy mark that you can get in this. If you get one of these questions, you should be really happy. So if you've got one bacterial cell and it divides 8 times, how many times or how many cells will be present after the 8th division. It's just the idea that you are going to be multiplying it by 2 each time. So 1 cell becomes 2 in the 1st division, 2 cells become 4 in the 2nd division, 4 cells become 8 in the 3rd division, 8 cells become 16 in the 4th division, 16 cells become 32 in the 5th division, in the 6th division it becomes 64, in the 7th division it becomes 128, and then it becomes 256 in the 8th division. I hope. Yes, does. God, it's almost like I'm a maths teacher,
Right.
isn't it?
Something that's important here, it's not your 2 times table, okay?
No.
So you should not be going 2 times 8 is 16 and then ending on that answer. The best way to do it is how it's written out here, okay? Definitely it's the longer way to do it. I'm sure there's something you can do with log calculators and actually...
Yeah. There is a maths way to do it, but just write it out.
Yeah, it's so much easier.
So much easier just to write it out. And each of those red arrows represents 1 division. You're not counting each of the green numbers as a division, it's each of the arrows saying this is 1 division, this is the next division, and so on to get up to it.
How many of those numbers have you got? Like, I just have the pattern memorized. Like, I can do 512 and then 1024, then 2048 and all that. I just have them memorized. You do not have to memorize them. Don't memorize them. It's a waste of your time. Okay, so there are some key terms that you're going to need to know before going into this. I promise they will make sense when we actually look at the process. But one key term is a chromosome is a strand of DNA that codes for all of the characteristics of an organism. That important bit is mainly strand of DNA. Okay, a chromatid is the name for a single strand of DNA in a duplicated chromosome. Okay, now we'll get into the idea of single versus duplicated chromosomes, but the idea is if you're ever looking at a structure inside a nucleus, it's going to be called a chromosome. It doesn't matter if it's single line or X-shaped, it's going to be called a chromosome. Okay, the equator, first big term bringing in there as well, is the middle position in the cell where the chromosomes are going to line up and attach to the spindle fibers during mitosis. Now that again is an important term. I promise it'll make sense in a couple of slides time. Spindle fibers are protein threads or fibers, and they are produced to pull apart chromosomes. Sorry, that's exactly what I shouldn't say. They are used to pull apart chromatids during mitosis and then pull chromosomes to the opposite poles of the cell. You see how I tripped up? I teach this fluently subject and I managed to trip over my language.
Don't do that.
Don't do that. Don't do that if you can help.
Right, so another idea that you need to get your head around before we go into the full process is the idea of something called the diploid chromosome complement. And that's the idea that in every cell, we have 23, or not in every cell, in almost every cell, I should say, we have 23 pairs of chromosomes, which is 46 chromosomes, because in pairs there's 23. And the idea is that this is, in humans, the diploid number. So 46 chromosomes is the diploid number. So 23 pairs, 46, and that is how many chromosomes we find in one cell. There are cells that this doesn't go with, and that's to do with your reproductive cells, which we will touch on in reproduction, but almost every cell in the body has this diploid chromosome complement, and that just means that they have 46 chromosomes present, and you do need to know that number. For humans, it's one that comes up a lot. It is different in different animals though, so it might come up in a question where it might talk about a rat or a pig or something, and it might say a different number as their diploid, and you just have to be aware that it just means that is the number of chromosomes in the cell or in the nucleus.
There is a little spelling mistake there. It should be complement, not complement.
Sorry, I'm dyslexic.
It doesn't matter for that. Okay, so mitosis. Let's have a look at this process then. So it's the process of cell division which helps maintain the diploid chromosome complement. Okay, so the idea is it's shown in the diagram in the front there a little bit. So you've got single chromosomes on the far left diagram. They are going to duplicate up, and they see how they form that X structure. The idea is there's now 2 chromatids in each chromosome. Okay, they line up along this line in the middle, they get pulled apart, they get pulled to opposite ends of the cell, and then 2 new daughter cells form. So we're going to have a look at those steps in more detail in the next couple of slides. Okay. So the first stage, essentially, of mitosis is that before mitosis, the chromosomes have to be doubled so each daughter cell can receive a full copy of the DNA. If you've got a chromosome number in this example of 2, okay, and the idea is you want to make 2 daughter cells, each with 2 in it, you can't say 2 divided by 2 is going to end up being 2. That's not a sum that works. 2 divided by 2 is 1. Okay, so instead what we have to do is double up the chromosomes, so you effectively end up with 4 copies of the DNA, and then split them in 2, and then you can end up with two daughter cells, each with 2 chromosomes. Okay.
And again, this is just a picture showing the same idea. The nucleus on the left contains 4 chromosomes. The nucleus on the right still contains 4 chromosomes. They're just replicated ones. So it's 4 pairs of chromatids, which is 4 replicated chromosomes.
Move on. Okay, so stage 1, I'll go through all the stages, but in an exam situation or in a test situation, you might not get a mitosis question that starts at stage 1. You might get one that starts at stage 3, or maybe the stage numberings are different, but the idea is this is the thing that happens first during mitosis. So even though they're not visible, the chromosomes make a duplicate copy of themselves. So the chromosomes are now X-shaped, made up of 2 identical chromatids, okay? So this is 1 chromosome in the picture with 2 chromatids that we can see as part of it. We don't say that there's 2 chromosomes there, that's wrong, okay? 1 chromosome with 2 chromatids.
In stage 2, this one's nice, the chromosomes now become visible. So they weren't visible before, they've then duplicated, now we can see them much more clearly. It's not a blurry mess anymore.
Okay, into number 3, the idea is the chromosomes are held inside the nucleus. The nucleus has a thing around the outside of it called the nuclear membrane. It's the thing that's keeping all the nucleus contents inside and stopping them escaping. So the nuclear membrane dissolves and the spindle fibers start to appear.
The chromosomes then line up at the equator of the cell, which remember that's that kind of imaginary line down the middle, basically just like there is on the Earth. And then each chromatid will attach to a spindle fiber. So the spindle fibers are at the poles, the chromosomes have lined up at the equator, and really crucially, it is each chromatid that attaches to a spindle fiber. Have to be really careful with your wording on this one. This is the one where everyone loses it because they say chromosomes attach to spindle fibers, but they don't. Each chromatid attaches to a spindle fiber.
The labeling on the diagram has gone slightly skew-whiff on this one, but the idea is the equator is the line where you can see all the X's lined up on. Okay, so the middle of the cell idea. The spindle fibers are those stringy things that you can see shooting out, kind of like Spider-Man's webs.
The faint grey bit, yeah.
Oh, there we go. That's the equator, spindle fiber, spindle fiber. Excellent.
You would have done that.
Okay. Spindle fibers then contract. Now, this is where your language needs to be super accurate as well. So the spindle fibers contract, and that pulls apart or separates the chromatids. But as soon as the chromatids are separate, they are now called chromosomes. Okay, so the chromosomes then move to the opposite poles of the cell. Okay, so while they're joined together, the chromatids then get separated apart. Chromosomes move to opposite poles of the cell. That's a very, very important language part. This sentence, ludicrously important. It seems like we're being overly picky, but we're really, really not. Okay, so here we have again our spindle fibers, spindle fiber. Okay, they sort of on this diagram, I'd say they'd actually be more called chromosomes. It would be more accurate for that. Because the idea is as soon as it's split apart, it's now a chromosome. It's only called a chromatid when they are joined together. Okay, so we can say chromatids are pulled apart, chromosomes move to opposite poles of the cell.
Then one of the final things to happen is a new nuclear membrane forms around. So remember, it's dissolved before, it's disintegrated at the start. So a new nuclear membrane now forms around this new group of chromosomes, and the cytoplasm begins to divide in 2. So we can get our nice 2 new cells basically pinched off.
So there's our new nuclear membrane. Okay, and that's the end of the process. 2 identical daughter cells have been produced. Each are also identical to the parent cell, so they have the right number of chromosomes inside the nucleus.
And this is a diagram. It'll come up more as well when you go on to the reproduction stuff, but the idea is obviously when we are formed, we start off sperm and egg fused together to form something called a zygote, which we'll touch on more in reproduction, but the idea is that is a single cell. It divides into 2 cells, it then divides into 4 and 8 and so on and so on and so on, and eventually, boom, you got a baby, essentially. There's a little bit more to that, but we'll touch on that later.
Okay, so key terms to know for this particular topic. So mitosis, it occasionally comes up in exam questions. It just asks you to name it sometimes. So it's division of a diploid cell to produce 2 identical daughter cells.
The spindle fibers, they are fibers that pull apart chromatids, and pull the chromosomes to poles of the cell.
The equator is the line that represents the middle of the cell. It's not a real thing, it's an idea.
Imaginary.
It's like the equator of the earth. You don't stand in the middle of the earth and see a giant line drawn in marker pen by Jesus. It's not there, okay? So instead it's just an imaginary line.
A chromosome is just a strand of DNA. It can be that single one, it can be the cross-shaped, doesn't matter, still a chromosome.
Okay, a chromatid is a single strand of a replicated chromosome that is attached to another chromatid. Very important.
The pole is just the end of a cell.
Okay, and the nuclear membrane is the membrane around the nucleus. Okay.
Okay, so to summarise, the things that you have to know, you obviously have to be able to name that process. We've talked about it 1,000,001 times now. It is mitosis. It is the process of which a cell divides.
Okay, you have to describe the stages of mitosis either from a picture or basically the next stage that would happen afterwards.
Yeah.
If you're confused, go back and listen to us talk through the 7 numbers again.
Yeah, and remember, they don't always come exactly as we've named them.
No.
It might be that what we've said is stage 1, 2, and 3 might be stage 1 for someone else. What matters is that you know it goes in this order. It doesn't matter about how it's numbered particularly, as long as you can say it like a story. It's like the difference between like a book and a film. Sometimes we miss little bits out, but the same idea is usually there, other than in terrible movies where it's not. But the idea is it follows the same pattern, the same things happen, as long as you know it goes in this order. That's what matters. So you also need to be able to obviously identify the stages from various pictures. This comes up a lot.
It really does. And then the very important, using that term chromatid versus chromosome correctly, equator and spindle fibers, and also maybe identifying them on a diagram of mitosis. Like they'll have a picture and say, what is part X? And that could be the equator, it could be spindle fibers, or maybe even a chromatid or a chromosome. Okay, so that's it for producing new cells. The next part of this key area is about stem cells, and then we'll finish off with cell hierarchy.
It's really nice.
We love stem cells.
Compared to mitosis.